Hello, everyone. Thank you all so much for joining today. We're going to be starting our seminar promptly at 1 p.m. Eastern Time or 10 a.m. Pacific Time. So it's in 5 minutes. Hi, everyone. Thank you so much for joining us today. We're going to be starting the webinar in about 2 minutes. All right, we're going to go ahead and get started. So hello, everyone. Thank you all so much for joining today. Welcome to the NOAA Omics Seminar Series. I am your host, Catherine Egan. I am the NOAA OAR Omics Coordinator, and I sit in NOAA's Office of Ocean Exploration and Research, or NOAA Ocean Exploration. I'm also the Executive Secretary of the NOAA Omics Working Group, as well as a subcommittee co-lead under this working group. So omics, which is a suite of tools used to analyze DNA, RNA, proteins, and small molecules, has become a large priority here at NOAA in the last few years. We established this seminar series in an effort to increase transparency and collaboration and highlight the incredible omics-related research currently underway with NOAA and its partners. The seminars from this series are all made available on YouTube and posted up on our omics website. There should be a link to our website in the chat right now. As participants, you all are muted, but feel free to type questions in the questions for staff box throughout the presentation. At the end of the presentation, I will read the questions for our presenter to answer. So with that, our presentation today is titled Implementing Genetics into Fisheries Management, Case Studies in Alaska Fisheries by Dr. Ingrid Spies. So Ingrid, thank you so much for presenting today. I'm going to switch screen sharing permissions over to you. And while you are getting situated, I'm going to introduce you to our audience. And also we can see your presentation just fine. So Dr. Ingrid Spies has worked at the NOAA Alaska Fisheries Science Center since 1991 when she started interning there in high school. She had the opportunity to go out to sea several times during college. She received her master's from University of Washington School of Aquatic and Fishery Sciences, where she used genetics to answer questions about the origin of sockeye salmon in Lake Washington. After working as a genetics lab technician at UW and the Alaska Fisheries Science Center, she pursued a PhD in the UW Quantitative Ecology and Resource Management program, focused on understanding the best tool to implement genetics into fisheries management and stock assessments. Ingrid currently works in the Status of Stock and Multi-Species Assessment program at the Alaska Fisheries Science Center. She works jointly on assessing stocks of Alaska groundfish and incorporating genomics to answer questions on fishery management. In addition to the assessment group, she feels lucky to work with a great group of researchers affiliated with the Auk Bay Genomics Laboratory, led by Dr. Wes Larson. So Ingrid, thank you so much for presenting today, and I'll go ahead and pass it off to you to begin.
Thanks so much, Catherine. I'm excited to be here. And I am going to be talking to you about an aspect of omics where I will be talking about applying genomics and also some transcriptomics to fisheries management of Alaska fisheries. This is a famous photo of me from a number of years ago, and it's quite possibly my most famous photo, holding onto a cod, which I will feature later in this talk. So as you all know, NOAA Fisheries is responsible for the stewardship of the nation's ocean resources and their habitat, using sound science and an ecosystem-based approach to management. My goal in this talk is to make the case that in line with NOAA's mission, omics benefits fisheries management, and I'll show you some diverse examples of this benefit. Thank you also, Catherine, for providing a definition of omics in the talk title. Obviously, it's a collective term and it includes many things: genomics, proteomics, metabolomics, and transcriptomics. And the goal is simply to quantify and characterize pools of biological molecules that translate into the structure, function, and dynamics of living organisms. So some of the ways I have listed here that omics benefits fisheries management, the first in my mind is the most obvious, identification of new species. If you don't know what species you have, you simply can't manage them. So I'll provide a quick example of that to start with. The second example, I believe, is slightly more nuanced, identifying management boundaries within species or their genetic population structure. And that's important so that in managed marine species, we assign appropriate management units. And I'll give you several examples of this. This might be the bulk of the work that I've done. And then the third example is better understanding responses to environmental covariates And for this, I'm going to use some examples through genomics and actually transcriptomics and examples of how we can better understand how fish and crabs respond to environmental stressors under climate change. And specifically, we can take this information into stock assessments and management through kind of a modeled covariate in the stock assessment. So most of this talk will be about genomics. And as you all know, genomics is an organism's complete set of DNA. And then within the genome, as you can see here, there are actually two types of DNA: mitochondrial DNA, that's located in the mitochondria, and this is often involved in metabolism. And we also sometimes, and this is, we sometimes use it for species identification. There are sometimes sections of the mitochondrial DNA that are unique to a species. Then there's genomic DNA, which you see here in the nucleus, organized into chromosomes, which is useful for comparative genomics, can be useful for population structure. So I think you can probably guess that I'm going to be talking about differences among and within species in the ocean. And I just included this slide to talk about the fact that barriers in the ocean exist, even though you might not intuitively think so. The ocean seems pretty like a homogeneous habitat in some sense, but there are definitely oceanographic, thermal boundaries, even sometimes being really physically distant acts as a boundary. And then sometimes there are boundaries that we don't exactly completely understand. For example, this is a really cool paper that I've read several times where they talk about how range limits in marine species can be mediated by flow. And they show the oceanographic flow in this map. And I point out just a couple of examples where Point Conception and Cape Hatteras are examples where you see breaks in species distribution simply because of oceanography and flow. So my first example of, and then I wanted to point out also that we have barriers to species distributions, probably due to flow and other oceanographic features in Alaska. So off Alaska, of course, we have the Aleutian Islands chain. And then as you see in this upper map, and then in the upper panel here, I love this plot by George Hunt in this 2005 paper, but it shows, almost lost them. There's my mouse. So it shows the Aleutian Islands chain, and it shows flow through these passes. The Aleutian Islands is really characterized by some large oceanographic passes. And then on the bottom panel here, it shows the depth. And so particularly, I'm just going to point out that Mchika Pass is the deepest pass within the United States portion of the Aleutian Islands. So a few years back, I was lucky to work with a couple of taxonomists, Jay Orr and Dwayne Stevenson and Jerry Hoff, who noticed that a particular type of skate, the Alaska skate, seemed to have a really particular coloration pattern in the western Aleutian Islands. And they noticed this and started wondering, what is going on? And working with them and using genetics, specifically a stretch of the cytochrome oxidase subunit 1 portion of mitochondrial DNA, which we mentioned earlier, can be used to distinguish among species in some cases, we were able to pinpoint that surprisingly there is a break in the species distribution of Alaska skate. And so east of Mchika Pass is your standard Alaska skate, which is distributed here in the eastern Aleutian Islands up through the Bering Sea shelf. And then west of Mchika Pass, it actually was this other species called the leopard skate. So to me, that was a really cool, very simple example of how to use omics in fisheries management. It's just at a simple level, how genetic data can assist in fisheries management. And so I'm just going to move on to the second way that omics can assist in fisheries management, which is genetic population structure. And there are many calls, as you see in these two quotes below, to match biological units with management units. for example, Linda Lakra wrote, sustainable management should be based on knowledge of genetic population structure to avoid depletion of genetic resources. And you see this quite a bit, especially by geneticists trying to make the point that their work is important. But, you know, just because it was said, wasn't necessarily conclusive evidence. And so for my PhD, I did several simulation studies and management strategy evaluations. I have to warn you, Google Scholar tells me people don't read these very often, and really they're pretty dry. So just listen, I'll give you the nutshell version. So the simulations and MSE showed that there are definitely benefits of using genetic data to make management decisions and incorporating genetic results, even given, you know, possibility for error, which you do in simulation studies, you always incorporate error. It is definitely significantly advantageous to incorporate those genetically driven management units, because it will reduce the possibility that you will unintentionally overharvest in some regions. And coincidentally, it will also increase your yield, as this plot shows. I use this example called catch cascading, where you actually precautionarily divide it among smaller management units, and that provided the best yield in these simulations. So just want to put that in to show you that, yes, there's evidence that doing this will be good for management, and it is based on sound science. So now that we've established that, I just want to give you a little more background before we move into some more studies on using genetics for identifying population structure. So within all species, all individuals share the same genes and the same number of chromosomes. But individuals within a species will have many genetic differences. And sometimes it's just random variation, such as maybe between siblings. But when you have groups that are separated, say maybe with little or no migration, their genomes will begin to diverge. Maybe it's randomly, or maybe for reasons to do with natural selection and local adaptation. And just taking a closer look, DNA, you know, it's a long molecule, and it's made up of 4 distinct nucleic acids, which we just refer to as A, C, G, and T. And I'm going to be referring to SNPs a little later on. And when cells divide, it's possible that there might be copying errors, where instead of a C, in this example, you might get a G. And so a SNP is simply a site along the genome where there are differences, and you might compare among individuals, or you might compare among populations. So one more point is that in the human genome and in fish and crab and et cetera, only a small portion is actually coding for genes, and the rest is non-coding DNA. And we sometimes will discriminate among these. We'll use it all in different ways. And I might get back to this point later on. So as you can see, there are different patterns that you might observe when you're looking for genetic population structure. On the left panel here, you could have distinct populations. You can have isolation by distance, which is kind of like a decline in allele frequencies rather than a sharp break. Or on the right, you can have kind of a random pattern, which is no differentiation. So these are kind of the things that you might look out for. Now, as Catherine mentioned, in addition to being a population geneticist, I work as a stock assessment scientist at the Alaska Fisheries Science Center. So I really get the opportunity to think and strategize about genomics projects that can benefit important questions in fisheries management. And this is a list of species that we manage in actually just the Bering Sea and Aleutian Islands. And there's a whole other long list for the Gulf of Alaska. But this is a lot of species. And so for each of these, we set quotas. A stock assessment scientist will run models. We'll set quotas. People will keep track of the catches. And so there's a lot going on. And my point in kind of bringing this up is that there's a lot of species to know what the genetic stock structure is and make sure you're your management units are appropriate. And so this is kind of a project that I and Dr. Wes Larson have taken on to kind of understand the stock structure, just to make sure that we're doing it properly with all these various species we assess. And also, there are some really interesting questions that come up that genetics and genomics and transcriptomics can help answer that I'm about to describe. So I'm going to go back about 10 years to a population structure project in which I used 17 microsatellite loci. And I didn't mention what this is, but it's simply a point along the genome, which describes variation, could be within or among populations. And my goal was to look for whether there were significant genetic differences between cod from the Aleutian Islands and the Eastern Bering Sea. And indeed, I did find that there was significant differentiation. And what was really cool that I think is a big takeaway in this kind of like interface between genetics and management is that in 2013, using this and other information, the Management Council actually split the management unit. It was previously one big management unit for cod between the Bering Sea and the Aleutian Islands, but then subsequent to 2013, the Aleutian Islands got its own management unit. And I'm kind of highlighting cod in a way. First of all, it's one of the most valuable species we assess and manage, but it's also, we've noticed that it's somewhat susceptible to climate warming. As Steve Barbeau, who now does the Eastern Bering Sea stock assessment, recently wrote a paper on from the Gulf of Alaska, they have declined as a response to ocean warming. And another interesting point about Pacific cod is that although we do summer surveys, we know they move into maybe different locations in the winter, and they're also fished at least as hard in the winter as the summer. So there's a lot of really complicated issues with cod and also with the fishery. But subsequent to the split, where the Aleutian Islands became its own management unit, I just wanted to point out what the stock size looked like. And it had actually declined through about 2010, 2012. And then it kind of did pick up just a little bit. And it's possible, and like I said, it's complicated, but stock size might have come back a little bit once it was managed as its own unit. So moving on, about 10 years after that microsatellite study, I was able to work with a group of researchers at the University of Washington, NOAA, and elsewhere to ask a similar question, the question, what is the difference between cod in the Bering Sea and the Aleutian Islands, or is there, using another methodology called pool-seq. And this is one of the next generation suite of sequencing that uses, that will give you millions of SNPs. So it's way better, way more informative than 17 microsatellite loci. It gives you a lot of information. The drawback is it does work by pooling groups of individuals that are from the same location, and that can be a drawback, but it does make sequencing more cost effective. What we found is really cool, and I'm actually pretty excited about it. So I want to point out this set of plots. Along the x-axis, there's kind of an upper panel and a lower panel here. Let's focus on the upper panel. Along the x-axis, you see numbers, and these numbers represent chromosomes. So cod has 23 chromosomes. And then on the y-axis, you have average FST, and FST is a measure of genetic variation, so that these peaks show regions of higher genetic differentiation. The black and gray represent loci or SNP locations that are not outliers, and the red are outlier loci. And then you might also notice these peaks. There's 8 or 9 of them here, and we call these islands of differentiation. So you have 9 peaks here in the upper panel that are representing islands of differentiation between the Eastern Bering Sea and the Aleutian Islands. Now let's look at the bottom panel now that we've established what this is. And the difference here is that instead of looking between the Eastern Bering Sea and the Aleutian Islands, you're now looking at differentiation between the Eastern Bering Sea and the Washington coast. And the difference between cod from the Eastern Bering Sea and the Washington coast is that in all the years we've ever tagged cod, we've never found one that's moved from the Bering Sea to Washington coast. So we don't think there's gene flow there. And you can also kind of, there's a good takeaway from this. You see there's much higher FST in this bottom panel because for whatever reason, which is probably genetic drift, these populations are not subject to gene flow, they've drifted apart. And in many cases, you see the same kind of peaks here. But in the upper panel, in places that are not these peaks, you have lower levels of FST. So what we think is going on is that you might have some regions that could be subject to local adaptation in these outlier regions, but then probably gene flow is acting to kind of homogenize Eastern Bering Sea and Aleutian Islands cod in the regions that aren't outliers. And we think that this potential adaptation could be due to the different environments that we have between the Aleutian Islands and the Eastern Bering Sea. These two regions are really different, both in depth, flow rates, and light availability and prey. So there's a lot of different things going on there. There's a lot more to learn. And probably what the tool that we need to learn more is, I mentioned with PoolSeek, we had to pool all the individuals together, so you definitely lose some information. So what we need is individual-based low coverage whole genome sequencing. And luckily, I'm so lucky to be working with Sarah Schall. She is a postdoc who is working with myself and Wes Larson, and she has done low coverage whole genome sequencing of Pacific cod at the locations you see highlighted here across all the regions they spawn in Alaska. And what you'll see here is what's called a PCA, which is a plot that shows relatedness among individuals. So each dot represents an individual, and it is color coded by its location of origin. And you can see clustering, like for example, the purples kind of cluster, and these are Eastern Gulf of Alaska samples. And the goal that Sarah is currently just about completed is to develop an informative panel of SNPs, not take all the millions that she has, but take only a selected few and come up with what's called a GTC panel. And on the bottom, I see something that might be familiar to you. This GTC panel is going to be used similar to the genetic tests that we as humans can send in the mail and figure out what you're related to. So that's kind of the goal. And this is kind of the groupings that we expect to see out of the GTC panel. The Bering Sea, you see clusters together, Western Gulf, Aleutian Islands, Eastern Gulf of Alaska. So this is really exciting in my mind, and I think it has a ton of potential. It can rapidly and inexpensively address several questions that are directly related to management of Pacific cod. Just in the next week or so, we're going to analyze a really cool set of age 0 cod that were captured in the Western Gulf of Alaska by Alyssa Abelkire, to identify if the spawning stock of origin differs between cod that have settled in different bays, and see if that differs among years. Another application that I'm very excited about is to learn more about the proportion of cod caught in summer surveys that were actually spawned in the area they were encountered. This is going to greatly help our stock assessments by understanding, is the is the summer biomass that we get really indicative of what's going on in the winter, or is it different? Are there a lot of Aleutian Islands fish that move into the Bering Sea in the summer and then move back to the Aleutians in the winter? And finally, well, not finally, actually, I think there are many more applications, but we will also use this to give some genetic basis, not genetic background, to the tagged cod that Suzanne McDermott's group has been working on. And they have lots of samples, and we've been able to actually analyze some of them. So I'm going to go over some initial results from some cod that were tagged in the, mostly in the northern Bering Sea, and I think it was in 2019. And so the orange and yellow samples you see here are cod that were tagged, and there were actually, they clustered in two different ways. So there was a group of them, of the orange and yellows, that clustered with Aleutian Islands samples. And I have to say, this is Sarah Schall's analysis. I'm giving her full credit, and she gave me her slides. And then I'll go over what the Aleutian Islands cluster did. So here on the left panel, you can see where they were tagged, mostly north in the Bering Sea, but also at least one was tagged in the Aleutian Islands. I bet that's right by ADA. And then in the right panel was their capture location. And it actually, we have the month of capture location on the right, and you can see that they were not all captured during spawning season. And they moved all over. There's one here at Petrel Bank that was recaptured in, it looks like May. One more in the Aleutians, also captured right by ADAC. Maybe that was the one that was released by ADAC. And then some other ones were captured in the Bering Sea. And then here's kind of zoomed in on that same plot of where these cod were captured and when. And then this other cluster of cod that were tagged in the Bering Sea that also appeared to have been spawned in the Bering Sea, they were found all over the Bering Sea when they were captured. And this plot shows the different months. And some of the months that you would expect them to be spawning, like February and March, it does look like they were along the eastern Bering Sea shelf, where we know that a lot of cod congregate to spawn there. But all this is really great information for understanding where cod go outside of spawning season. And so I'm going to highlight a study that we did without a GTC panel, but what I really want to make the point of is that a GTC panel, we were lucky to be able to do this without this panel, but if we had it, it could rapidly and quickly help answer questions in the short time period, possibly even between a survey and an assessment season. So in 2017, we found a surprising, people who were on the race survey found a surprising number of cod that had moved into the northern Bering Sea from their last survey there in 2010. And it seemed to possibly be related to a lack of a cold pool. We feel that, or we've noticed, researchers have noticed that cod movement appears highly affected by the cold pool in the eastern Bering Sea. They can be in that water, but they do tend to avoid it. And just to kind of give you an idea of how much the biomass changed, it increased almost about an order of magnitude from 2010 to 2017, and concurrently it really decreased in the eastern Bering Sea. So this was a big thing, and we were kind of like, oh man, what are we going to do? But luckily, we had genetic baselines from 6 spawning populations, and we were able to take those. Oh, and luckily we also took samples of those northern Bering Sea fish, and we were able to characterize them. And this again is another principal components analysis where you see points are individuals and the clusters represent spawning populations. And you can see sort of, they're kind of overlapped, that the northern Bering Sea samples overlapped exactly with Unimac and Pribilof. There's one more in there, Unimac and Pribilof spawning samples. And so this result was able to provide really important information to the stock assessment scientists that their stock hadn't all died, it had just moved. and the shift in cod biomass prompted the use of northern Bering Sea survey data in the assessment. So that was a really helpful one. And it was, yeah, so we hope to be able to provide information like this in the future. Okay, so similar to Pacific cod, we, meaning my group, and Ellie Bors was the one who actually prepared the libraries and selected the samples with me. We have performed low coverage whole genome sequencing of walleye pollock. And I have to give credit also to Eleni Petru, who did quite a bit of bioinformatics, and now Sarah Shaw, who's also working on bioinformatics, and we will at some point prepare a GTC panel. And what we found with walleye pollock is that similar to cod, there was a surprising level of genetic stock structure. So really, we have a lot to learn about this species, and it will lead us to better understanding of how to incorporate stock structure into the management of it. Now, finally, I want to get to the last way that omics can help in fisheries management, and that is with transcriptomics. Over the last quite a few years, the Alaska Fisheries Science Center has been conducting a series of experiments on the effects of temperature and ocean acidification on fish and crabs. This work has been done in a laboratory setting in Kodiak with Chris Long, and also at the Newport Lab with Tom Hurst and Ben Laurel and Emily Schlesinger. And oh, this slide I actually missed. So I wanted to cover this as another kind of a background point, which is that when DNA, or when genes in the DNA are turned on to synthesize protein, RNA is transcribed. So studying the RNA that is expressed when individuals are subjected to different conditions can give you a hint and a clue at what genes are transcribed or expressed. And it is really appropriate for a laboratory study to understand the response to a stressor. And so I'm going to describe to you an experiment on red king crab. And we know that they are sensitive to ocean acidification. Oh, and I wanted to give all the credit actually to Laura Spencer, who's our bioinformaticist on this project. She prepared these slides and did absolutely an amazing job. And we know that crab are sensitive, particularly at the juvenile stage. And there's been quite a bit of experimental work by Chris Long and others that have found high mortality and decreased growth when we expose juveniles to ocean acidification conditions. But we don't exactly know why or the mechanisms behind this decreased growth and increased mortality. It's not clear how exactly ocean acidification affects their physiology of crab or fish. So in this particular experiment that I'm describing, red king crab were reared to the larval phase, from the larval phase to the juvenile phase, and they were subjected to 3 different treatments: an ambient, which is a pH 8, you'll see in a few minutes, moderate acidification, which is pH 7.8, and severe acidification, pH 7.6. And what we found, I'm just going to give you everything here, what we found is that crab reared in higher ocean acidification conditions are more physiologically constrained. We see less gene expression, less variability among individuals, fewer biological functions are active, and we actually see larger effects at more severe ocean acidification. And this PCA kind of demonstrates how we have this variability in expression. So you see the crab in red. These are individuals again that were reared in the severe OA conditions. See, they cluster quite closely together, which means that their genes are expressed at similar levels in all crab, whereas the crab reared in ambient conditions don't cluster quite as tightly, and their expression profiles were much more diverse. So they had more broad gene function. So that was one of our significant results. But then the next question is, well, what kinds of questions were less active in these crab that were subjected to severe OA? There were quite a few and quite a variety of processes that were actually downregulated in OA reared crab that included energy production and metabolism, protein synthesis, you see here in translation. Their immune system was downregulated. That is not a good sign. And the TCA cycle means the Krebs cycle, which is the main source of energy for cells. So there are quite a few downregulated things. And actually, downregulation of the immune system is particularly interesting because it makes us wonder, does OA make red king crab more susceptible to infection? It might not kill them directly, but it could make them less capable of fighting off other pathogens that they encounter, particularly in ocean warming. And there were actually much fewer processes that were upregulated. But what it did include was regulation of transcription, the nervous system that you see here in axonogenesis. There was negative control of growth. And finally, one thing that was very interesting was this transposition, which has to do with transposable elements. And those are also called jumping genes, or parts of the genome that'll insert themselves in certain places, which we don't totally understand, but in other organisms, we know that they are implicated in stress response. And in this case, it's not completely we're not completely understanding if they're helpful or harmful, but this is a really, really cool, interesting thing. Now, overall, we have found that OA limits the breadth of the physiological functions in crab. It results in decreased survival and growth, specifically in terms of energy and metabolic limitations, and it suppresses the immune system. And then we have this high transposable element activity. So yeah, this is pretty bad news for red king crab if the levels of OA become more severe in the future. But the good news for stock assessment is that it provides really sound science behind a strong covariate towards mortality, and that we could use this information to understand and quantify this relationship between OA and mortality. So one more point I wanted to make is that eDNA is another aspect of omics that can be useful for management, but I'm not covering it. And I just wanted to kind of introduce Anna Betcher, who works at the Ock Bay Genetics Lab, and maybe she'll provide more information. I think she is doing excellent work on eDNA that I'm not going to cover. And so there's probably even more people to thank, but many people have helped in all of this research, funders. So I think everybody, even if they're not listed here, who has helped over the years. And with that, I'm ready to take questions.
Awesome. Thank you so much, Ingrid. That is a fantastic presentation. While the questions trickle in, so folks feel free to ask questions in the questions for staff box, and we'll read them out loud to Ingrid. But while those trickle in, I did want to point out that Ingrid highlighted a ton of collaborators here, and many of these folks have actually given seminars as part of the NOAA Omics Seminar Series. Dr. Larson, Dr. Shaw, and Dr. Betcher have all given presentations. So if you go to the Omics website that's linked in the chat, you can see recordings of all of those past presentations as well. So we're happy to take any questions from the audience. So I think maybe we'll just wait a moment and see if some questions trickle in. I think what happened is that Ingrid, you did such a great job with your presentation that everybody was like, no questions this time around. Okay, we do have one that came in from Lauren. So Lauren asked, this fantastic work is following single species for stock assessment. How can this be wrapped into ecosystem-based fishery management? Are there any multi-species studies to define the geographic extent of different ecosystems?
Wow, great question, Lauren. Multi-species. I think, for example, with the last example I gave with crab, I mentioned that this was just one of several crab species that we will be doing a similar suite of experiments on. So after we finish with red king crab, we'll be doing tanner and snow crab. And that might be a nice little case study. So if there was, for example, a multi-species model that incorporated multiple species of crab, you could look at differential response of these different species to OA and have the same kind of relationship, but perhaps different variance or effect by species. So that would be one. Did that answer that? Well, I guess I can't talk back and forth with you, Lauren, so we'll follow up later.
Thanks, Ingrid. Yeah, Lauren, if you did have like a follow-up question, you could just go ahead and type it in the chat and I'll read it out loud when we get there. Okay, well, our next question is from Sandy, and they ask, in the collections for Sarah, for the individual genome expression, was maturation status of individual fish taken into account and the collections time relative to the spawning season? Just wondering about the spawning phenology of stocks as another barrier.
Yeah, great question, Sandy. Yes. So for all the fish that we sample, we only collect during spawning season. And sometimes it's a little tricky depending on who collects for us to get detailed information on whether they're actively spawning or not. But generally we'll get spawning or non-spawning is the most basic. And so we sometimes do have to say, we collected these cod during spawning season and they were all of mature size. And that's sometimes the best we can do. But in cases where we have more information, actually, Mary Fisher in particular did a great study on this where she was able to see that there were some anomalous fish in a spawning group, and she was able to use maturity and actually it was size that implicated maturity to say that these fish were not actually spawning there. So yeah, we do our best to get maturity information, and when it's available to us, sometimes it can be used to draw some pretty interesting conclusions. Thank you.
Awesome, thanks, Ingrid. Our next question is from Daniel. Are there similar activities or research with omics occurring at Alaska Fish and Game for salmon management? And then he says, great set of studies presented today.
Thank you. Was that a question or it was a statement that similar cool things are being done at ADF&G?
I think it's a question. Just wondering if maybe something's being done at the state level.
I think so. I'm pretty sure. I know some individuals that work in those organizations, but I particularly can't speak to them. But Daniel, if you wanted to email me, you can, and I can put you in touch with people I know in those organizations and you can ask.
Excellent. Thanks, Ingrid. All right, our next question is from Franz, apologies if I pronounced your name wrong. Are there efforts at Alaska Fisheries Science Center or other centers to use genomics for estimating population abundance, e.g. close kin mark recapture?
I think, well, I know that Eric Anderson, who's at the Southwest Fisheries Science Center, has done a lot of work on this. And this is definitely, obviously, something we're so interested in doing, but the constraint is sampling enough young fish to have sibship and half sibship well quantified, that it's really kind of been a barrier. If you can imagine, you know, the size of, say, for example, the cod population in the Bering Sea, and then you can imagine, given the fecundity of the cod population, how rare it would be to find sibs or half sibs in a random sample. So that's really been the issue that we're up against, is that the probability of that is so incredibly low that, so doing this close kin mark recapture, we haven't found it to be like practical at this point. Now I know they do it in some other stocks where they have it worked out. I have even actually looked for sibship in some of my results, and I haven't been convinced that this would be something that would be like worth going after in any of the species that I've looked at, although I would really like to. And just to kind of like give the audience an idea of what we're talking about, this is, again, it's a way of sampling individuals and using some complicated analyses to get at what is the stock size, how many fish are there. So it's really cool, it's really tempting, but it also, there's definitely some things that make it tricky.
Great, thanks, Ingrid. Okay, we have a few other questions that are coming in. This next one is from Paul, and they say, can this work help the management of multi-species complexes? In that case, the issue is not really stock structure, but rather that there are multiple species managed under a single management unit.
Oh yeah, that is really tricky. And I just have to tell you, Paul, that as a stock structure or like a population geneticist, I really kind of cringe when we have multiple species that are managed under in a stock complex, because you really can't do much for one of these species. One of the species could be vulnerable under that kind of lumping. I guess I should explain for the audience that sometimes with species that are caught together, for example, say sculpins, there's like 50 species of sculpins in the Eastern Bering Sea, and they're all just managed under one group. So actually not even in the management plan anymore, but at one point they were. So that means if you catch a certain biomass of sculpin, that just counts towards the overall sculpin quota. But say you were catching all of the butterfly sculpin only, and you actually might be catching really too many butterfly sculpin, it would totally fly under the radar, because there's really no way to protect the butterfly sculpin since they're only managed as just a group of all the sculpins together. So yeah, so I've kind of said that managing as a complex makes me a little nervous, but on the other side, I understand the reality is that there's no way that any fisherman or observer could tease apart all these 50 different species of sculpin. It's just not practical whatsoever. So I get that too. So there's the practical aspect, and then there's the kind of the more finicky science aspect, and they all work together to balance each other out, and it works out.
Thanks, Ingrid. Okay, our next question is from Nikolai. It appeared that most of the stock ID work you showed tended to result in identification of substructuring and potentially reasons to divide a single stock into multiple stocks. Have you had experiences where the genetic work showed that stocks managed separately were not genetically distinct? And if so, has the management of those stocks changed to assess and manage them as a single stock?
Oh, that's such a great question. And that actually leads to and I think, Nikolai, I think you're right. I mostly did highlight cases where there were differences because I thought it would be maybe more interesting. I didn't want to show you all a lot of examples where we looked for stock structure and didn't find it. But yes, they definitely exist. And then you're struggling with, what do you do with that information? Because that's also really, really, really important information. So here's an example. Atka mackerel in the Aleutian Islands is a very important fishery. These are schooling fish, so they're found in big and caught in big groups. And quite a few years ago, we did a study to look for genetic stock structure, and we found absolutely nothing. And that was fine. But what's interesting is with Atka mackerel, we actually precautionarily still manage them in smaller groups than not in the whole Aleutian Islands. If you saw that one of the fisheries management maps I put up earlier actually divides the Aleutian Islands into Western, Central, and Eastern, and Atka mackerel are managed precautionarily in each of these groups separately. And so even though the genetics found nothing, no reason, no genetic differences, that precautionary management by NIMS area never changed. And I think if it's not a big hindrance to the fishery, not a big hindrance to the people that manage it, then it's okay. And so that is an example. That's a cool example that I'm glad you brought up. And yeah, thank you.
Great, thanks, Ingrid. Okay, I think we have one more question here, and then I have a couple questions that I think I need to answer. So this one comes from Joseph. Are there plans for adding transcriptomic data to population structure in cod and pollock? If so, which tissue would be more informative for transcriptomics?
I thought about that because we are doing transcriptomics work on cod currently. And actually, our first attempt was with cod that were so very, 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 very small that we decided to just analyze the whole cod, which is okay, but it's better to have particular tissues. And I think the tissues we decided on that would be most valuable for cod were the head or the brain region and I think it was the upper kidney. And I can't off the top of my head remember the exact reasons for that. But if you email me, Joseph, I can get back to you and tell you exactly why we selected those. And it's true, different parts of organs are more informative than others when you're doing this kind of work.
Excellent. Thank you so much, Ingrid. I think that was all the questions that we had for you. I did have a couple people add to the chat to answer Franz's question earlier about close kin mark recapture. And they're saying that, yes, this is also done at other fisheries science centers here at NOAA. And then I also had a question that came in that said, will the recording be available after the presentation? Yes, absolutely. Please bookmark the website link that's currently in the chat. This is where I link all of the recordings from this seminar series. So with that, I want to just take another moment to say thank you so much, Ingrid, for presenting today for the seminar. As stated, you gave an excellent presentation, and we really appreciate you being here today for this.
Thank you.
All right, I gotta do a little wrap-up spiel for everybody here. So I also want to thank the audience for joining us today. The NOAA Omics Seminar Series takes place on the third Wednesday of every month at 12 p.m. Eastern Time, although sometimes we do have these seminars scheduled outside the time. So once again, just check the website for up-to-date seminar information. Our next seminar is on September 21st and is titled Using Metabarcoding to Understand Microbial Communities on Aquacultured Sugar Kelp. Presented by Dr. Yuan Liu. So I hope to see you all there. Check out the Omics website for registration details. All right, and with that, I'm going to end our webinar. Thanks so much for joining today, everyone.